Hello everyone, welcome to Get My Class. Today we're going to solve this question which says match the following where they have given column X and column Y and they have given A denaturation, B extension, C annealing, D initial denaturation. And they have given column Y, that is 40 degrees Celsius, 95 degrees Celsius, 55 degrees Celsius, 72 degrees Celsius. We have to match column X and Y and further we have to look for correct option from A, B, C, D. So first, let us talk about the first one, which says denaturation. So what do you mean by denaturation? Denaturation is a process by which the double-stranded DNA will get separated into single-stranded DNA. And usually this happens between the temperature 44 to 94 to 98 degrees Celsius. In some books, they have mentioned 96 to 98. So nothing to worry. If you have to talk about the optimum temperature, it will be 95 degrees Celsius is the optimum temperature. So that is what is denaturation. So the first one will match up with the second option. Okay, the first one will match up with the second option. The B says extension. So what is extension? The extension is nothing but addition of the nucleotides. Addition of nucleotides so as to increase the length of the DNA. Okay, that is what is extension, or you can also say synthesis of the DNA which occurs, and this is brought about by Taq polymerase in case of PCR. Now, what is PCR? That is polymerase chain reaction. So the steps what we are discussing here is actually the steps which occur in PCR, though it is not in the correct order, because whatever they have given according to the column we are going to discuss. So actually, according to PCR, the first will be denaturation, then it will be annealing, then you will be having extension and further termination. Okay, so here it is extension, that is nothing but addition of nucleotides, and this is done by Taq polymerase in case of PCR. And this Taq polymerase is thermostable enzyme, which is a polymerase enzyme, and this will perform very well at 72 degrees Celsius. This is the optimum temperature. So the extension part will match up with option 4. Okay. Now the C1, it says annealing. So what is annealing? Annealing is nothing but binding of the primers to the primers to the DNA strand. So the DNA, the single stranded template. Okay, DNA which is acting as a template. Okay, and this happens when the temperature is little lower than the extension temperature, that is around 55 degrees Celsius. Okay, so 55 degrees Celsius, that is the optimum temperature. Hence, annealing, you will say it is third one. Then D says, what does D say? D says initial denaturation. So, what do you mean by initial denaturation? This again, initial denaturation is again nothing but the separation of the double-stranded DNA. Initial denaturation. Here also there is separation of the DNA. Okay, so double-stranded DNA will get converted into what? It will get converted into single-stranded DNA. Okay, this again the optimum temperature is 95 degrees Celsius only. So the D1 is also the fourth one. Okay. Sorry, this is the second one again, because as I told you, it is 95 degrees Celsius. So where is 95 degrees Celsius? Here. So we can say that the correct option will be the first one, that is denaturation is second one, that means at 95 degrees Celsius. Extension occurs at fourth one, that is at 72 degrees Celsius. Annealing will occur at third one, that is at 55 degrees Celsius. And initial denaturation again will occur at 95 degrees Celsius only. Okay, so that means for both denaturation, initial denaturation, it will be second one only. So let us see. Option A says A is 2 correct, B is 4 correct, C is correct, and D is also correct. Okay, so it is correct according to our option, that is whatever we have got, 2, 4, 3, 2. 2, 4, 3, 2. That is absolutely right. Option B says A is 4, that is, it says denaturation occurs at 72 degrees Celsius, which is ruled out. Option C says A is 2, correct. B is 1, it says. That is annealing, the extension occurs at 1, that is 40 degrees Celsius, which is incorrect. Option D says A is 2, B is 4, that is correct. C is 3, that is correct. And D is 1. It says initial denaturation occurs at 40 degrees, which is incorrect. So the correct option is option A. Let us conclude. What is the answer? The answer is option A. I hope you understood the explanation. Thank you.